Dear students, a warm welcome to all of you for today's discussion. The topic we are going to discuss today is fluorescence in situ hybridization, or in short form fish technique. The main objectives of this discussion are to make you understand the definition of fish technique, steps involved in fish technique, various types of probes used, protocol for fish technique, applications of fish technique, different approaches for future development of fish technique, disadvantages or negative aspects of fish technique. Now let me start the discussion with the introduction of fluorescence in situ hybridization, or fish technique is a cytogenetic technique developed by biomedical researchers used to detect and localize the presence or absence of specific DNA sequences on chromosomes. In the year 1969, Pardue and Gall first described the in situ hybridization of radioactively labeled repetitive DNA probes on cytological preparations. In 1986, Pinkel and Creamer reported fluorescence in situ hybridization using non-radioactively labeled fluorescence. The high sensitivity and specificity of this technique and the speed with which the assays can be performed have made fish technique a powerful tool with numerous applications, and it has gained general acceptance as a clinical laboratory tool. Fish is often used for finding specific features in DNA for use in genetic counseling, medicine, and species identification. It supplements conventional karyotyping by providing additional information in certain cases. Fish to localize genes on chromosomes. Fluorescence in situ hybridization can localize a gene to a specific place on a chromosome. First metaphase chromosomes are isolated and attached to a microscope slide. The chromosomal DNA is denatured into single-stranded pieces that remain attached to the slide. The fluorescent probe hybridizes to the corresponding gene, and when the slide is illuminated, the hybrid molecules fluoresce and reveal the location of the gene of interest. Now next we go to the important steps used in fish technique. They are slide preparation and DNA denaturation, nucleic acid hybridization, and visualization of the probe by using fluorescence microscopy and digital image analysis. Slide preparation and DNA denaturation being the first step, and in this step, cells in metaphase or interphase are fixed on a slide and denatured to change the double-stranded DNA into single-stranded DNA. Nucleic acid hybridization is the second step. Nucleic acid hybridization is the formation of a duplex between two polynucleotide chains which have complementary sequences, that is DNA-DNA, DNA-RNA, and RNA-RNA. The metaphase or interphase preparation is then hybridized with DNA sequences that are complementary probe to the region of interest and that have been labeled with biotin. The hybridization site is made visible by means of a primary antibody against biotin. This antibody is bound to a fluorochrome, for example, fluorescein isothiocyanate or FITC. Since the primary signal is quite weak, a secondary antibody, for example, avidin bound to biotin is attached. A further primary antibody can then be attached to the secondary antibody. This amplifies the signal, which can then be demonstrated by bright fluorescence. Visualization of the probe by using fluorescence microscopy and digital image analysis. DNA probe is labeled with a colored fluorescent molecule attached to the DNA during the hybridization process. The molecule emits a particular color when waved through a fluorescence microscope that is equipped with the appropriate filter sets, CCD camera, digital camera systems incorporating a variety of charge coupled device sensor photodiode are by far the most common image capture technology employed in the modern optical microscopy. Now the important probes and fluorescent dyes that are used for fish technique. Probe is a nucleic acid that can be labeled with a marker which allows identification and quantification. Probes are the complementary sequences of target nucleic acids designed against the sequence of interest. Probes are tagged with fluorescent dyes like fluorescein, biotin, and deoxygenin. The differences between the various fish techniques are usually due to variations in the sequence and labeling of probes and how they are used in combination. Probe sizes range from 20 base pairs to 1000 base pairs. Probe size is important because longer probe hybridizes less specifically than the shorter probes. A short strand of DNA or RNA which is complementary to a given target sequence can be used to identify or locate the target. Now, direct and indirect labeling. What is direct labeling? Fish probes are labeled directly using fluorochrome conjugated nucleotides, for example, fluorescein DUTP. Label is bound to the probe, less sensitive. Indirect labeling requires an additional step before detection, indirectly using reporter molecules, for example, biotin DUTP, by nick translation, random priming, PCR, or various other molecular genetic techniques. Probe detected using antibodies conjugated to labels like alkaline phosphatase results in amplification of the signal. Fluorescent signals of bound probes are inspected using filter-equipped epifluorescence microscope and computer software. Specificity of probes. A large number of different probes designed to identify specific chromosomes and parts thereof are available for diagnostic purposes. The choice of probe and the simultaneous use of multi-probe assays depend on the particular application in question. One of the most important considerations in fish analysis is the choice of probe. A wide variety or range of probes can be used, from whole genomes to small cloned probes. There are broadly 4 types of probe, each with a different range of applications. First is whole chromosome painting probe WCP and partial chromosome probe. The second type of probes used in fish technique are repetitive sequence probes and telomeric probes. They are specific for telomeres. Third types of the probe used are locus specific probes or locus specific identifiers. Now the fourth type of probes are centromeric probes or chromosome enumerator probes, whole chromosome painting probe WCP and partial chromosome probe. Routine application of the technique on human chromosomes started in the year 1996 with the simultaneous use of all 24 human whole chromosome painting probes in multiplex fish and spectral karyotyping. Since then, different approaches for chromosomal differentiation based on multicolor fish, M-fish, assays have been described. Different names have been introduced for more or less the same probe sets. Multiplex fish or M-fish, spectral karyotyping or SKY, multicolor fish, M-fish, cobra fish, 24 color fish. M-fish and SKY allow painting of the entire chromosome complement in a single hybridization through labeling each chromosome with a different combination of fluorophores. Images are collected with a fluorescence microscope that has a filter set for each fluorochrome, and a combinatorial labeling algorithm allows separation and identification of all chromosomes, which are visualized in characteristic pseudocolors. Multiplex fish and spectral karyotyping differ only in the method used to discriminate the differentially labeled probes. Spectral karyotyping uses a dedicated imaging system that incorporates a cooled charge coupled device, CCD camera. Multiplex fish uses specific narrow fluorescence filter sets to reduce crosstalk and digital imaging equipment as part of a conventional epifluorescence microscope with appropriate computer software. The high efficiency of modern epifluorescence microscopes reduce typical exposure times, limiting the effects of photobleaching. The main applications of multiplex FISH have been chromosomal rearrangements and marker chromosomes in solid tumors, which are often distinguished by complex karyotypes. In common with other whole chromosome painting methods, both multiplex FISH and SCI are not suitable for discriminating intrachromosomal rearrangements such as duplications, deletions, inversions. The second type of probes used in FISH technique are repetitive sequence probes and telomeric probes. They are specific for telomeres. They hybridize to these specific chromosomal regions or structures that contain short sequences which are present in many thousands of copies, have specificity for a single human chromosome arm. They contain a locus estimated to be within 300 kb of the end of the chromosome. Examples of this probe type are pan-telomeric probes targeting the randomly repeated sequences present on all human chromosome ends. In most instances, these sequences are distinct, such that a satellite probe derived from one chromosome will hybridize to that chromosome only. Third types of the probe used are locus-specific probes or locus-specific identifiers. Interphase FISH can be used for the detection of any chromosome abnormality for which there is an appropriate probe. Probes of this class are particularly useful for detecting structural rearrangements such as specific chromosomal abnormalities like deletion. Deletion probes, translocation probes, gene detection and localization probe. Gene amplification probes, usually genomic clones, which vary in size depending on the nature of the cloning vector. For example, a locus-specific probe for the BCR, breakpoint cluster region, gene at 22q11.2 detected with a green fluorochrome. A locus-specific probe for the ABL, leukemia virus, which carries a similar protein, Abelson oncogene, gene T9q34, detected with a red fluorochrome will appear as a bright yellow spot. The combination of green and red fluorochromes in leukemia cells when viewed via fluorescence microscopy, characterized by the BCL or ABL fusion gene, resulting from T9:22, Q34.1:Q11.2. This is a reciprocal translocation, creating an elongated chromosome 9, termed a derivative chromosome or derivative 9, and a truncated chromosome 22, the Philadelphia chromosome. In agreement with the International System of Human Cytogenetic Nomenclature (ISCN), this chromosomal translocation is designated as T9:22, Q34:Q11. Now the fourth type of probes are centromeric probes or chromosome enumerator probes. For chromosome enumeration, probes detecting chromosome-specific alpha satellite regions near the centromeres are preferred, generated from repetitive sequences found in centromeres. Most are alpha and satellite 3 probes. Centromere regions stain brighter means they are rich in AT bonds. Centromeric probes which target all human centromeres are also available. Satellite DNA probes hybridize to multiple copies of the repeat unit present at the centromeres, resulting in a two very bright fluorescent signals in both metaphase and interphase diploid cells, hence making centromere specific probes particularly suitable for the detection of monosomy, trisomy and other aneuploidies in both leukemias and solid tumors. A centromere specific probe for chromosome 8 has been used to detect trisomy by FISH in newly diagnosed patients with acute myeloid leukemia. Molecular beacons as a fluorescent probe. A molecular beacon is a fluorescent probe molecule that is designated to fluoresce only when it binds to a specific DNA target sequence. This probe has two engineered regions at the ends of the probe sequence. One on the 5' side, a fluorescent tag is added, and on the 3' side, a Kenshin group is added. DNA sequence of 20 to 30 bases. The central region of the probe is complementary to the target sequence. The terminal half dozen bases at each end of the probe are complementary and form a short double-stranded region, and in the stem and loop of conformation, the Kenshin group is next to the fluorophore and so prevents fluorescence. Just inside the two tags are 6 base pairs that can form a stem loop structure. In this state, the probe cannot fluoresce. When the probe binds to the target sequence, the stem loop structure is lost. Since the Kenshin group is no longer next to the fluorescent tag, the probe can now fluoresce. When the molecular beacon binds to the target sequence, it is linearized. This separates the Kenshin group from the fluorophore, which is now free to fluoresce. Technical improvement and recent development of fish technique. This is what we are going to discuss now. Centromere-specific multicolor fish. Recently, on all human centromere-specific multicolor fish approach for the ascertainment of the origin of structurally abnormal, cytogenetically unidentifiable chromosomes, so-called marker chromosomes. This technique allows simultaneous characterization of all human centromeres using differently labeled centromeric satellite DNA as probes. Comparative genomic hybridization. One of the most significant developments for fish in relation to genome-wide screening was introduction of comparative genomic hybridization, or CGH. This modification of quantitative two-color fluorescence in situ hybridization utilizes genomic DNA from the sample under test to generate a map of DNA copy number changes in tumor genomes, making it an ideal tool for analyzing chromosomal imbalances in tumor material and for examining possible correlations between findings and tumor phenotypes. In CGH, the genomic DNA from the specimen and control DNA extracted from an individual with a normal karyotype, that is 46XX or 46XY, are differentially labeled with green and red fluorochromes respectively, mixed in equal amounts and co-hybridized to reference human metaphase chromosomes. The relative difference in DNA content and between the normal specimen DNA is represented by difference in the green-red fluorescence ratios. Virtual karyotype. Virtual karyotyping is another cost-effective, clinically available alternative to fish panels using thousands to millions of probes on a single array to detect copy number changes, genome-wide at unprecedented resolution. Currently, this type of analysis will only detect gains and losses of chromosomal material and will not detect balanced rearrangements, such as translocations and inversions, which are hallmark aberrations seen in many types of leukemia and lymphoma. Now, what's the protocol for fish technique? The reagents and equipment necessary for this technique are 20x saline sodium citrate buffer, RNase A 100 micrograms per ml in 2x SSC, pepsin 40 units per ml in 10 millimoles HCl, paraformaldehyde, ethanol, labeled probe, hybridization mix solution, wash buffer, that is 20% formamide in 0.1x SSC, detection buffer 0.2% tween 20 in 4x SSC, blocking buffer 5% bovine albumin in detection buffer, antibody or detection compound, DAPI 2 micrograms per ml in antifade mounting medium, fluorescence microscope with filters, glass slides, plastic cover slips for incubation and hybridization steps, heat block, modified thermocycler. Now, slide preparation. Chromosome preparation from any cell type. Incubate with 200 microliters RNase for 1 hour at 37 degrees centigrade. Then wash slides in 2x SSC for 5 minutes and repeat. Rinse slides in 10 millimole HCl Incubate with 200 microliters pepsin for 10 minutes at 37 degrees centigrade. Rinse slides in deionized water. Wash slides in 2x SSC for 5 minutes and repeat. Stabilize slides in paraformaldehyde for 10 minutes. Wash slides in 2x SSC for 5 minutes and repeat. Dehydrate slides in an ethanol grades 70% followed by 80% and finally 95%, 2 minutes each. Then air drying of the slides is done. Now third step is hybridization. Heat slides to 70 degrees centigrade for 10 minutes and place on ice. Secondly, add 30 microliters of hybridization solution on each slide and cover with plastic cover slip. Denaturation of slide at 65 to 70 degrees centigrade for 5 minutes on heat block. Gradually decrease temperature to 37 degrees centigrade. Hybridize at 37 degrees centigrade overnight in humidity chamber. Detection of cytogenetic abnormalities under fluorescence microscope. Wash slides in 2x SSC to remove cover slip. Wash slides in wash buffer at 40 degrees centigrade for 5 minutes and repeat. Wash slides in 0.1x SSC at 40 degrees centigrade for 5 to 15 minutes. Wash slides in 2x SSC at 40 degrees centigrade for 5 to 15 minutes. Cool slides to room temperature. Equilibrate slides in detection buffer for 5 minutes. Block in blocking buffer for 20 to 30 minutes. Incubate with 50 microliter antibody or detection compound for 30 to 60 minutes. Then wash slides in 2x SSC for 5 minutes and this is to be repeated twice. Counterstain with DAPI solution for 10 minutes. Rinse briefly and mount in anti-fade mounting medium. Then is observation under fluorescence microscope. By now we discussed about the definition of fluorescence in situ hybridization, steps involved in FISH technique, important probes and fluorescent dyes that are used for this technique, the technical improvement and recent development of FISH technique and the protocol for the technique. Now we will move on to the various applications of FISH technique. The high sensitivity and specificity of FISH and the speed with which the assays can be performed have made FISH a pivotal cytogenetic technique that has provided significant advances in both research and diagnosis. FISH technique has greatly expanded the capabilities of cytogenetics and pathology laboratories through its high sensitivity, specificity, and rapid turnover with a high efficiency of hybridization and detection. The broad applications of FISH technique are diagnosis of genetic diseases. From a medical perspective, FISH technique can be applied to detect genetic abnormalities such as aneuploidy, loss of a chromosomal region or a whole chromosome, or to monitor the progression of an aberration, serving as a technique that can help in both the diagnosis of a genetic disease or suggesting prognostic outcomes. Examples of diseases that are diagnosed using FISH include cry-du-chat or cat's cry syndrome, also known as 5p- syndrome. It is a chromosomal condition that results when a piece of chromosome 5 is missing. Infants with this condition often have a high-pitched cry that sounds like that of a cat. Second is the Down syndrome. Down syndrome results from trisomy 21, which means each cell in the body has 3 copies of chromosome 21 instead of usual 2 copies. Then velocardiofacial syndrome. Velocardiofacial syndrome, also known as VCFS, as 22q11.2 syndrome or as Di-George syndrome, is the most common syndrome associated with cleft palate. Velocardiofacial syndrome is an autosomal dominant condition. Genetic studies of children with this condition show that a microscopic segment on the long arm of chromosome 22 is missing. One more is Prader-Willi syndrome. Prader-Willi syndrome is a rare genetic disorder in which 7 genes or some subset thereof on chromosome 15, Q11, 13 are deleted or unexpressed. Chromosome 15q partial deletion on the paternal chromosome. Fish technique can also be used for biological species identification. Fish technique is also used in clinical studies. If a patient is infected with a suspected pathogenic bacteria, the patient's tissues or fluids are typically grown on agar to determine the identity of the pathogen. Many bacteria, however, even well-known species, do not grow well under laboratory conditions. Fish technique can also be used to compare the genomes of 2 biological species to deduce evolutionary relationships, known as zoo blot. Fish technique is widely used in the field of microbial ecology to identify microorganisms. Biofilms, for example, are composed of complex, often multi-species bacterial organizations. Preparing DNA probes for one species and performing FISH with this probe allows one to visualize the distribution of this specific species within the biofilm. Preparing probes in 2 different colors for 2 species allows to visualize, study co-localization of these 2 species in the biofilm and can be useful in determining the fine architecture of the biofilm. Detection and measurement of mRNA levels. A variety of different mRNA species are expressed at any one time within a tissue. Not all cells express the same genes or express them at the same level. If the target cells are probed with fluorescently labeled DNA red, this will bind to the corresponding mRNA blue. Notice that the probe DNA does not bind the nuclear DNA because in this procedure, the cells were not treated to denature the chromosomal DNA. The target gene in this example was only expressed in 2 of the cells. The different approaches for the future development of FISH technique. Recent advances such as improved hybridization protocols and strategies, novel commercial and non-commercial probe sets, alongside the efforts of the Human Genome Project, coupled with hardware development such as image analysis, software and specific filter sets for newly established fluorochromes, facilitate the collection of new important data for the understanding of genetic diseases. Now, before we conclude the discussion, let me tell you some of the disadvantages or negative aspects of FISH technique. FISH and to a lesser extent SCI and mFISH can only detect known genetic aberrations, provided the specific probe is available. FISH analysis with locus-specific probes on chromosome-specific DNA libraries is restricted to the target chromosomes or chromosomal subregion. FISH technique cannot serve as a screening test for chromosomal rearrangements since most FISH techniques can only detect known imbalances. mFISH and SCI are not useful for distinguishing intrachromosomal rearrangements such as duplications, deletions or inversions. Having said about the various disadvantages of FISH technique, I conclude the discussion by mentioning here that despite minor disadvantages, FISH has proved invaluable in both diagnostics as well as research. The power of its ability to identify specific genetic aberrations has propelled FISH-based techniques to the forefront of screening procedures for prenatal, pediatric and adult cases in a wide variety of cell types. The ultimate goal of FISH utilization would be an array-based DNA nanotechnology screen to diagnose any prenatal or postnatal chromosomal aberration. This was all about the FISH technique. Hope you got the topic well. Thanks for your patient hearing.